Okay, let's unpack this. We're diving into the absolute core of life today, the genetic code.
Right.
For decades, the foundational concept, the one taught in every biology class, was the central dogma of molecular biology. It's the blueprint for how instructions flow.
That's right. And when Francis Crick first articulated this idea back in, the late 1950s, he framed it as the fundamental rule for information flow in the cell.
And the simple textbook version, often attributed to James Watson, was wonderfully clean. DNA makes RNA, and RNA makes protein.
It's a nice, simple, linear path.
It really is. And it's the skeleton of how we think about heredity.
But if you, our listener, are still relying solely on that simple linear model, you are missing the entire orchestra playing around that skeleton.
The orchestra, I like that.
Our deep dive mission today is to move far beyond that simple flow. We're going to explore the intense, dynamic layers of control, the regulatory feedback loops, and the vast complexity that truly orchestrates how and when genetic information is expressed.
Okay, so before we jump into those complex controls, let's quickly set the stage with the language itself, the genetic language.
Yeah, the building block.
It's an incredibly efficient language. Proteins are built from just 20 different types of amino acids. And to code for them, the cell uses a sequence of 3 nucleotides, a codon.
Right, and mathematically that gives you 64 possible codons.
With 61 actually coding for amino acids and the others acting as, stop signals. It's precise, but it really needs to be.
And that precision, it all starts with that deterministic flow. We still rely on the 3 core sequential steps. First, you've got replication, that's DNA copying itself, so DNA to DNA. Second, transcription, where DNA is read to make an RNA intermediate.
And then finally, translation.
Finally, translation, where that RNA message is used to build the protein.
So let's talk about transcription, that DNA to RNA step. It's run by RNA polymerase, or RNA pol. This enzyme just zips along the DNA template, building the RNA strand in the 5' to 3' direction.
And what always fascinated me is the difference between the 2 core polymerases.
DNA polymerase versus RNA polymerase.
Exactly. DNA polymerase, the replication engine, needs a little starting flag, a primer, to get going. But RNA pol, it doesn't. It just knows where to start.
Yeah, it just goes.
And that seemingly minor difference, it hints at the core function. DNA is the master copy. It has to be perfect. So DNA polymerase is a very careful proofreader.
But RNA pol isn't.
Not nearly as much. It has much lower fidelity. It makes errors roughly once every 10,000 to, say, 100,000 nucleotides.
So why is that tolerated? I mean, why is the RNA copy allowed to be so much messier than the master DNA?
Because the transcripts are temporary, right? They're meant to be used, often locally and quickly, and then just destroyed. If a flawed transcript makes a flawed protein, the cell can just scrap the transcript and make a new one. The core blueprint is never damaged.
It's an economy of speed, really.
An economy of scale and speed, exactly.
And this is where eukaryotes, complex life, including us, add a huge layer of complication. In bacteria, transcription and translation are basically linked. They happen at the same time.
Right, the RNA is being translated into protein almost as soon as it's made. But we separate them. DNA is safe in the nucleus. Protein synthesis happens out in the cytoplasm.
And that physical separation demands a whole lot of processing of that initial RNA copy, the primary transcript.
The pre-mRNA, yeah. Before it can even leave the nucleus, it has to be modified. It gets a protective 5' cap and a long poly A tail on the other end. But the real game changer in eukaryotic complexity is splicing.
Splicing is where the cell acts like a truly meticulous editor. The pre-mRNA is full of the actual coding sequences, the exons.
The pure instructions.
Interrupted by these non-coding junk sequences called introns. Splicing is the process of precisely cutting out every single one of those introns.
And then seamlessly rejoining the pure exons.
And the reason this just blows up the complexity is something called alternative splicing.
By mixing and matching which exons are included or excluded from that final mRNA, the cell can create multiple totally distinct functional proteins from a single gene.
To really grasp the scale of what you just said, we used to think, one gene, one protein.
The old model.
But if you look at the human DSCAM gene, which is crucial for wiring our brain through alternative splicing, that one gene can generate over 38,000 distinct protein versions.
38,000. It completely demolishes that old simple ratio. It fundamentally changes what a gene even means. And, that massive complexity also underscores the fragility of the whole system.
Oh, it's genuinely frightening to think about the precision required. I mean, look at the reading frame. We saw that example of just a single base pair insertion mutation.
Just one letter inserted.
And when that template is transcribed, the RNA sequence shifts the reading frame for every single codon after that point.
The change is catastrophic. You shift an original sequence, let's say MET LYS THR, into something completely unrecognizable, like MET LYS TRP his Lihu.
You haven't just changed one amino acid.
No, you've created a completely different protein, almost certainly a non-functional piece of molecular garbage. It's precise, but so vulnerable to error.
Okay, so if the core mechanics are already this complex, we have to ask, where does the dogma, that simple linear flow, break down entirely?
Right. Moving into the exceptions.
That's Act 2.
Crick's limit? It pretty much holds. Protein information can't flow back to nucleic acid. No. But that RNA step is incredibly fluid. We know about reverse transcription RNA being copied back into DNA.
Which is what retroviruses like HIV use to get into our genome.
Exactly. And we also see RNA viruses that skip DNA entirely, just replicating RNA directly from an RNA template.
But then you highlighted a really strange case that challenges the boundaries even more. Intanes.
Intanes. Yeah, these are segments of a protein that essentially edit themselves out after being made.
Wait, they splice themselves out of the protein?
It's a process called protein splicing. The protein folds in a specific way after translation and performs a self-cleavage reaction, cutting itself out. But what makes this a real exception is that many of these excised intanes can then go on to edit the DNA sequence itself. They contain specialized domains that can initiate DNA double-stranded break repair, which allows them to effectively edit the underlying DNA.
Hold on, is that a major functional pathway, or is it more of a rare sort of parasitic mechanism for the intent to propagate itself?
It seems to be mostly a selfish parasitic element, yeah. But its existence is proof that proteins can influence the underlying genetic sequence through direct action on DNA. It just challenges the assumption that the flow is universally unidirectional away from the protein.
Okay, so if that's the strange exception, the true revolution has been the sheer volume and function of non-coding RNAs, or ncRNAs.
Oh, absolutely.
LNcRNAs, mRNAs, circRNAs, they were once just dismissed as transcriptional noise, cellular filler.
We thought of them as noise because they don't produce proteins, so they didn't fit the simple dogmatic endpoint. But we now know they are the conductors. They function as scaffolds, catalysts, and incredibly complex regulators. Their impact is so widespread, we have to rethink that simple linear model.
And to really understand why this RNA-based orchestration is so critical, we need to look at a model system that just demands complexity. The neuron.
Exactly. I mean, think about a standard cell like a liver cell. If it needs a new protein, it makes the mRNA in the nucleus, ships it out, translates it. Simple. Now think of a motor neuron. Its cell body is in your spinal cord, but its axon might extend a meter down your leg. If that synapse at the end needs a rapid local protein change, say to strengthen a memory, it can't wait hours for a message to be transcribed in the nucleus and shipped all the way down.
So the neuron requires gene expression that's rapid, highly localized, and plastic.
It has to be. Relying only on the nucleus is just too slow and inefficient for a cell with that kind of architecture.
Which necessitates this huge system of RNA-based regulation and local translation right there at the synapse.
The RNA is trafficked to the site, ready to go on demand. And this brings us to the complexity of the RNA interactome itself, starting with just diversity.
The numbers are staggering.
Over 100,000 long, non-coding RNAs, LNcRNAs, are recorded in humans. The vast majority of them still have uncharacterized functions. We don't know what they do.
And the second key challenge is their structure. Unlike proteins, which tend to fold into one stable form, RNA is plastic. It can shift between multiple conformations.
And that structure, or lack of it, defines its function. A great example of this scaffolding is the LNcRNA called NEAT-ECI1. It's essential for forming these little compartments in the nucleus called perispeckles.
Like storage units.
Exactly, storage units for specific regulatory proteins and transcripts.
And then there are circular RNAs, circRNAs, which are really common in the brain. They don't have a start or an end. They're a continuous loop. Why make a loop?
Because the loop makes them incredibly stable, and they can act as molecular sponges. Take one called CRS7. It's basically a sponge for a specific microRNA, miR7.
So it soaks up the regulator.
It just sequesters that microRNA away from its real mRNA targets. It's a way to tightly control gene expression simply by locking up the controller itself.
That's incredible. So even once a functional mRNA is transcribed and spliced, the control mechanisms are still running full steam.
Oh, absolutely. We're now in Act 3, dealing with chemical marks and final protein adjustments. These post-transcriptional toggles make sure the mRNA is stable, goes exactly where it needs to, and is only translated exactly when needed.
Let's look at alternative polyadenylation, or APA. This process determines the length of the tail end of the mRNA, the 3' untranslated region. Why does the tail length matter so much?
Well, that 3' UTR is the regulatory hub of the RNA. It's where microRNAs and various proteins bind. So by generating versions with different UTR lengths, the cell changes which regulatory factors can even attach.
And longer UTRs.
Longer UTRs often act like a molecular zip code, targeting those transcripts all the way out to distant synapses for local translation. It's a key mechanism for rapid distance signaling in neurons.
And beyond just length and location, there are chemical marks placed directly on the RNA itself. The big one in research right now is N6-methyladenosine, or M6A.
Think of M6A like a highly specific postal stamp placed right on the RNA.
It doesn't change the message itself.
No, but it tells the cell's mail carriers, we call them reader proteins like YTHDF1, exactly where to deliver it.
So that one little stamp dictates the transcript's fate. Immediate nuclear export, maybe rapid translation,
Or maybe straight to the recycle bin for decay.
Yeah.
Precisely. And these dynamics are being shown to be tightly linked to neuronal plasticity and consolidating long-term memories. You're literally tweaking the stability and translation of key memory genes with these chemical tags.
And then finally, we get to the protein stage, and the modifications still continue. Post-translational modifications, PTMs, these are chemical edits to the proteins that completely transform their function.
Phosphorylation is probably the most famous one. It's a crucial molecular switch. A kinase adds a phosphate group, a phosphatase removes it. On, off.
And we see the tragic failure of this switch in neurodegenerative disease. In Alzheimer's, tau hyperphosphorylation, too many phosphate groups, causes the protein to fall apart and form toxic tangles.
But it can also be protective. In Parkinson's, PINK1-mediated phosphorylation activates a protein called parkin, which is critical for clearing out damaged mitochondria from the cell. Essential maintenance.
Another fascinating pair of PTMs you mentioned involves these small protein modifiers, ubiquitination versus assimilation.
Right. Ubiquitination typically tags a protein for swift degradation. It's the cellular destroy signal.
Get rid of this.
Assimilation, on the other hand, often does the opposite, promoting greater protein stability.
But stability isn't always good, right? We see in clinical studies that assimilation can actually promote the toxic aggregation of key disease proteins.
Exactly. It promotes aggregation of tau in Alzheimer's and alpha-synuclein in Parkinson's. These tiny molecular decisions, add a phosphate, add a sumo tag, have massive pathological consequences.
All of which brings us to the modern challenge of genomics. When we sequence a patient's genome, we see thousands of variations. How do we tell the difference between a driver mutation that causes disease and a passenger change that's harmless?
It's a huge needle in a haystack problem. Experimentally testing every single mutation is just too slow and way too expensive. And this is where computational biology has had to step in.
To predict the impact of these mutations.
Exactly, without ever entering a wet lab.
So what does a tool like MCSM actually see? Is it modeling the biology or the physics of the structure?
It's modeling the structure. Tools like MCSM use machine learning and graph-based signatures to represent the protein's environment. They basically predict, based on physics and chemistry, whether swapping in a new amino acid will destabilize that structure. And then tools like DUT enhance that prediction even further by combining structural models with statistical approaches.
And these tools are so essential because, again, the problem isn't just in the protein coding sequence. We're finding that SNPs, single nucleotide changes in non-coding RNAs, can be profound drivers of disease.
That's right. A single letter change in a non-coding region can be devastating. We see specific SNPs in LNCRNAs like HOTAIR linked to autism and schizophrenia, and mutations in MELA-T1 associated with multiple sclerosis.
So they don't change your protein recipe.
No, they alter the LNCRNA's structure or its expression level, and that completely throws off these complex regulatory networks in the brain.
Which means if non-coding RNAs are a major problem, maybe they're also the most promising solution. You highlighted targeting natural antisense transcripts, or NATs.
NATs are LNCRNAs transcribed from the opposite strand of a protein coding gene, and they often regulate that gene negatively. So by designing a therapeutic to silence the antisense transcript, you can potentially boost the expression of the healthy functional protein.
And the proof of concept is fascinating. In a mouse model where the antisense transcript for brain-derived neurotrophic factor, or BDNF, was deleted.
They saw a boost in the actual BDNF protein levels.
And that wasn't just on paper.
No, dramatically enhanced memory function in the mice. If we can use this antisense tech to boost BDNF, a key molecule for memory, just by silencing its counterpart. This opens up a totally novel path to treating memory loss.
One that targets regulation, not the protein itself.
Exactly.
What a journey. We started with the linear central Dogma DNA to RNA to protein, which we now understand is really just the structural skeleton.
And we end up here with a dynamic, fluid, highly orchestrated system where non-coding RNAs are the conductors and chemical marks and splicing events constantly adjust the score in real time. This level of specialization, particularly in neurons, is what makes the genetic code so much more than a simple blueprint.
And it leaves you with this provocative thought. Given the highly specialized architecture of the neuron and the vast, uncharacterized diversity of these non-coding RNAs, we've only met a fraction of them, what novel ncRNA, located in a single isolated synaptic compartment, is yet to be discovered that acts as the ultimate master regulator for long-term memory encoding?
The initial code may be cracked, but the orchestration is ongoing, and it's still full of surprises.
Exactly. That's it for this deep dive. Thank you for joining us. We'll catch you next time.